All right, so today we're going to reconstitute some PCR primers that we got. So we ordered these. They come as a lyophilized stock, so freeze-dried. So they come in these little tubes. Depends on where you order them, perhaps. First thing we do is label each of the tubes on the cap. Let's see if I can show you that. Just write the name on the top there. That's going to be useful later when these end up in a freezer box and we're trying to find them a year from now. We'll be able to look down and see the name. We prepare all of our primer stocks to a 200 micromolar concentration. When we get the data sheet or product sheet from the company, I'll show you here, it lists all the properties of the primers, how much we get. This column here will tell us if we want to make a 100 micromolar stock. So for the first one here, it says 200, we would want to add 207 microliters. Remember, we don't want to do that. We want a 200 micromolar stock, which is twice as concentrated as 100. So what we just do here is half the number that they provide. So here, if it says they wanted 207 to dilute this, to prepare this to 100 micromolar, we actually want half of that, so 103.5 microliters. So we just jot all those on there. We'll dissolve these freeze-dried primers in TE buffer. You can find the recipe in a lab book for TE buffer. This one is dedicated just for reconstituting primers, like to keep that separate. Obviously, if you contaminated these primer stocks when you're making them, that's going to have downstream problems where all your PCR reactions potentially could be contaminated. And so there's information when you get these products that'll describe how to reconstitute the primers. Basically, we add that volume of TE buffer, let it sit for 5 or 10 minutes. That allows that lyophilized stock to, those primers to dissolve into that solution, vortex it a couple times, and then from that we'll make our working solution of the primer. So I'll add those volumes of TE buffer and we'll look at how to make a working solution after that. All right, so we're ready to add our TE buffer to our dried primer stocks. So the first one I'm doing here, I need 103.5 microliters. Get that adjusted correctly. And this is obviously pretty straightforward. We're just adding TE buffer to tubes. But one thing I always do is use a new pipette tip for every one of these. When we're dispensing something, if we're careful, we could open the tube dispense, and if we don't touch the pipette tip to anything, potentially we could use that same tip. But because this is so important as an early stage in a lot of different PCRs, I just play it safe. We use a new pipette tip every time. So I'll continue to do that for each one of these. Once I've added it, I'll vortex these, and we'll let them sit for 5 minutes, and that'll be it. Okay, so our primers are resuspended now. Got them in our tubes here. It's a better view. We have our original stocks. These are resuspended to 200 micromolar. But that's not going to be our working stock. These are the things that are going to end up back end up in the -80 for long-term storage. We pull them out as needed. But we're going to make working stocks at 50 micromolar. These are the ones that we'll have in the -20 here in the lab that we'll use routinely. And then when we run out, we'll have to go back to the -80 and make some new stocks. So we label these with the primer name, and then we also write 50 micromolar. So it's really clear that's the concentration of the primer, and that's the standard working concentration of primers that we use in the lab. Different labs might do that differently. The key is, write on it, say what it is. And then it can be helpful if you consistently make your primers to the same concentration. That way, primer for one assay, you know it's at 50 micromolar, just like the primer for some other assay. So there's consistency. You don't have to think about it every time you're running a new PCR. There's that consistency. All right, so we're going from a 200 micromolar stock to a 50 micromolar working solution. That's a 1:4 dilution. All right, so to make our 1:4 dilution, we'll use 75 microliters of TE buffer. That's already in the tube here. And then add 25 microliters of our original stock that we just dissolved. So they're ready to go. So that 25 microliters in 100 total, so 25 plus 75, that's a 1:4 dilution. You can always do the math ahead of time if you're not sure. Always check, double check. But that's a 1:4 dilution. So we're taking something that's 200 micromolar, and diluting it down to 50. And like we do with a lot of things in here, as we do each one of these, forgot if I used that pipette tip. As we do each one of these, we just shift the tubes up in the rack. So we just move them up a notch as a reminder, okay, we've done that one. We move on to the next one. So it's helpful to do that. All right, so I'll finish these up, and that's how we make working solutions of our primers that we use in the lab.